So hello everyone, my name is Lizzie Stewart. We are pleased that you could all join us for this week's lecture in Volume 11 of our No Neuropsychology Didactic series that brings you lectures from experts in the field covering different topics each week. This series was created by trainees and early career neuropsychologists, including standing members of the No Neuropsychology Board, as well as members of the No Neuropsychology Committee. Before we start our lecture, one of the main goals of No Neuropsychology is to provide free, high-quality didactic content to our audience. Every No Neuropsychology and No Neuroanatomy lecture is available on our YouTube channel. Be sure to check it out, subscribe, and like our lectures to get first access to new content. Additionally, No Neuropsychology works to bring you learning and discussion questions that are provided with specific lectures content. You can access these on our website. Here are our disclaimers for the series. This training is not meant to replace formal education in neuropsychology, and the views of the speakers are their own. Questions can be submitted via the Q&A box in the lower left of the screen, and a recording of today's lecture will be provided on our website and YouTube channel later this week. Now it is my pleasure to introduce Dr. Emily Rogalski for today's lecture entitled The SuperAging Research Initiative, Identifying Protective Factors to Promote Healthspan. Dr. Emily Rogalski is a Rosalind Franklin Professor of Neurology at the University of Chicago, where she is Director of the Healthy Aging and Alzheimer's Research Care Center. She is a clinical and cognitive neuroscientist, and her research falls under the broad umbrella of aging and dementia. Using a multimodal approach, she focuses her investigations on two aging perspectives: primary progressive aphasia, which is a neurodegenerative disease that invades the language networks, and superaging, in which individuals are seemingly resistant to the deleterious changes in memory associated with normal or more typical cognitive aging. Her PPA research has helped to characterize the clinical and anatomical features of PPA, including drivers of disease progression, identifiers of risk factors, and refinement of our understanding in the language network organization. She has also pioneered a line of research concentrated on maximizing care and quality of life for individuals living with PPA and related dementias. She operationalized the superaging phenotype and has helped to establish the unique biologic, molecular, genetic, and psychosocial features associated with superaging. She currently leads the SuperAging Research Initiative, which holds promise for identifying protective factors, avoiding Alzheimer's disease, and optimizing health span, and reducing stigma and negative expectations associated with aging. So everyone, please join me in welcoming Dr. Rogalski.
Thank you, Lizzie. It's so wonderful to be here. I appreciate the invitation and the opportunity to share with you guys today. And I'm just going to take a second here to share my slides and screen. It was such a kind introduction, and I thought I would just spend a little bit of time on where I'm situated at the University of Chicago. We're within a new center called the Healthy Aging and Alzheimer's Research Care Center, or HARC Center for short. And this center was started in September of 2023 when I joined the University of Chicago. And we celebrated our first birthday back in September. And so we put some cartoon balloons on our picture here. And the SuperAging Research Initiative that I'll be talking to you about today is just one of those aspects of research happening within the HARC Center. So I hope that you'll spend some time getting to know us and the other research happening here. I'm excited to get to know all of you and would be happy to answer any questions that you have at the end. We'll start today with really kind of just an overview of superaging, what it is, what it isn't, and then some of our key features and findings to date. I'll start by saying we're really at the beginning of this story. We're still uncovering and getting to better understand these individuals who maintain great memory performance. So as we move on, I want to start with a big thank you to our funders, but also to our partners. And so you see those universities listed here. And I want to start by giving a big thank you to the huge team. It takes a true village to make this work happen. And as you see, there are many, many names listed here, and each one of them is contributing a tremendous amount to identify superagers, to do the research that you'll see more about today. So a huge thank you to them. So before we can really jump into what is super aging, I thought it might be helpful to take a step back and just talk about really broad strokes of simplified trajectories of aging. So when we think about aging, we tend to focus on the things that go wrong with aging. And that makes a lot of sense because they have a tremendous impact on our emotional well-being. They have an impact on our economy. They have an impact on our healthcare system. And so the trajectory that commonly comes to mind first when we talk about aging are pathologic trajectories of aging. This is where Alzheimer's disease and related dementias would fall because the biggest risk factor for these diseases is actually getting older. So merely age is that biggest risk factor for accelerated cognitive decline. And in this trajectory, there's severe restrictions in daily functioning. So the cognitive decline that happens is impacting daily life. There's another broad stroke or trajectory of aging called normal aging or average aging. And this trajectory is associated with some degree of cognitive decline but these individuals are able to maintain active engagement in life, so it's not interfering with their activities of daily living. And really, for years and decades, these were the only trajectories of aging that were talked about. And while that makes sense on average, and on average, we may see cognitive decline, if you start to think about the people you know, it doesn't always fit. So nearly everyone I talk to has a grandma, a grandpa, an aunt, an uncle, a neighbor who you say, "Oh goodness, you'd never realize you'd never know that this person was age 80, age 90. They run circles around me, or they're so busy I can barely keep up with them." This trajectory of aging started to be called. This concept of successful aging came into being from Rowan Kahn, and it started to get this construct of those who were doing well. But we had this idea to kind of take that one step further because successful aging didn't have just a single definition. It was just this idea of you could be age 65 and without dementia, and that might be a successful trajectory. Or living to age 100 could be another successful trajectory. We wanted to identify individuals and then study them scientifically to understand, could we find people who have little or no age-related cognitive decline? And so they're maintaining this high functionality in their later years. And so this was the theoretical construct with which superaging was, the idea of superaging was born. And then, of course, it needed some practical definition. And part of this was not just anecdotal, thinking about friends and neighbors that you might know, but if you looked within the literature, there's actually evidence of potential superagers. So now I've shown here across the x-axis, decades across the lifespan from age 30 to age 90. And then on the y-axis here represents memory scores, where higher numbers are better. And you see what happens on and then each of these dots represents individuals, whereas the asterisks represent kind of the average performance over time. And what you notice is that there is this average decline, but underneath it all, when you look at the dots, you see that there's quite a bit of variability. And then there's even these individuals that are represented by triangles here, where they seem to be doing as well, if not better, than individuals who are decades younger. So this gave us the idea that these individuals exist in addition to our kind of anecdotal experience with friends and neighbors. So the rules around superaging are as follows. We're looking for individuals who are over age 80. And you might say, oh gosh, well, why age 80? And part of that is because age is that biggest risk factor for changes in cognition. And if you live to age 80 and are able to maintain performance at least as good as individuals in their 50s and 60s, there is a steep decline that happens. So if you, for example, on the Ray Auditory Verbal Learning Test, the average 50 to 60-year-old needs to remember at least 9 words at the delay, compared to an average 80-year-old who only needs to remember 5. So you kind of get the opportunity to perform almost half as well and still be called average or normal. And we require that individuals have episodic memory performance at least as good as individuals in their 50s and 60s. Performance in other cognitive domains, such as language and attention, needs to be at least average for age, so not impaired. And these are the primary criteria for superaging. And then we invite these individuals to participate into a longitudinal and multidisciplinary study that allows us to look at factors that allow these individuals to maintain this great cognition over time. And eventually, we ask individuals to donate their brain at the time of death. And this gives us a special opportunity to look at cellular and molecular features associated with their clinical and cognitive phenotype. We enroll both superagers and cognitively average controls so that we have a built-in comparison group that has similar demographics and allows us for the exact same battery of tests and other evaluations. So before I kind of switch gears and start to tell you about the results and some of the things that we've observed to date, I thought it might be nice to put some names and faces behind the superagers, not just bar graphs and data. So we're going to spend the next few minutes here investing in meeting some of the superagers.
92 years old, and I hope to keep going, maybe slower and not as far, but I want to keep going, learning, and taking more adventures in.
I am 82 years young. I'm just having a ball. I'm grateful. And I want to be here a long time, like 100 and something.
Each day is another opportunity for me to live to the fullest in how I can help others, how I can enjoy myself, how I can be as creative and adventurous as I like being.
I think what makes me a superager is being active and positive.
As a result of the testing, I was asked to participate in the SuperAging program, which I was honored to do. So that's how I got involved. And that's what made me say, I'm a superager.
I didn't think I was really anything special until they said, you're super now.
I don't really feel super. I feel normal, and I'm happy to be 86 and be able to do all the fun stuff I like to do.
And now, 108, about 2 weeks from 109. And I can say, this has been a wonderful life. It's not all happy. It's not all sad. It's not all glad, but it's living.
60 was an awareness and acceptance of who I am. 70, woohoo, I'm still here. 80 was younger friends because I'd lost so many of my friends. And then finally I decided at 90 it was be mellow, cool it. You can't change it.
I have never been more relaxed and certain that I can control the 24 hours of the day as I want to spend it.
I joined this study purely out of curiosity, and I figured, oh, what the heck, maybe I can qualify and see where it takes me.
And I didn't know anything about superagers until I got a call and they asked me, and I said, yeah, why not? And I'm always curious about how my brain is working, and if it's working and how long it's going to work and, that sort of thing. So that's how I got into it. And I've met my peers. They're fun people. And I always believe in diversity, and it's always good because it broadens your understanding of others, other communities, other peoples, other lifestyles.
I was just asked to see if I wanted to be a superager, and I said yes. Anytime you use the word super, I'm in for it. The superager study experience, wonderful, because I have the bragging rights that I'm a superager.
A friend of mine who was in the study too told me about it. And I was a little skeptical because, you don't want to fail at something, but I thought it was worth a try. unfortunately, I was accepted for it and have really enjoyed the experiences and the opportunities it's given me.
If I wasn't here right now, I would be probably looking at the spring cleaning of my yard. I would be taking my bike down and getting it greased up, ready for a ride. And then I would be looking for a nice place to go to lunch that would have outdoor seating.
I would be in my car, driving up the outer drive, listening to my music and taking photographs.
I'd be in the pool swimming. And after 45 minutes of high intensity water aerobics, lifting weights, I would then walk a couple of miles and then I would take a yoga class.
Age is just a number. And to be a super ager, hey, that's cool. Doo-doo.
So hopefully that gave you a little snapshot of the super agers, who they are. They have so many important stories to share with us, so much wisdom to share with us. If I had told you that you were going to see a 108, almost 109-year-old today, is that what you would have pictured? I also want to share that Miss Edith is no longer 109. She's actually 110 and a half and doing quite well. We had the opportunity to visit her a couple months ago, and she was making homemade wine that she shared with us over the holidays. Miss June, who you saw in the video, she decided to go on a semester at sea last year with a bunch of college students. So these are active individuals who are really defying expectations and giving a new opportunity to look at aging. And so I think getting to know them is an important part, which is really a nice transition to why is it that we would study super agers? And I think there's a few different important perspectives to think about. One is that we've gotten better at extending our lifespan. So individuals are living longer, but it's sometimes coming at the cost where our health span is not keeping up with our lifespan. So there's this imbalance between the two things. We think superagers represent a situation where lifespan and health span are more balanced, more in sync with each other's. And so it gives us this opportunity to understand how do those things happen and how might that be helpful to others? How do we prevent disease and disability associated with chronic aging? We also think superagers give us the opportunity to inform about Alzheimer's disease. One way to study Alzheimer's disease is to study what's going wrong with the brain and then to try to reverse that. And that's certainly a laudable approach and one that has been very fruitful, but it's also taught us that Alzheimer's disease is extraordinarily complex. And when we have a complex problem like that, it's sometimes good to take more than one perspective. And so we need to continue this work of understanding how do we reverse or what has gone wrong, but it may be equally important to understand how do we prevent it to start with? Or how do some individuals tolerate that same Alzheimer's pathology, but it doesn't have the same impact? These are questions that the superagers help us answer. Are they resilient do they have plaques and tangles and they're resilient to them, or do they avoid getting them altogether? From a societal perspective, we think superagers give us a great opportunity to redefine expectations. So this is a picture of Miss Edith when she was, a young 102, and she was being interviewed by our local Chicago newspaper here. And during that interview, they did not only print photos, but they also did a video interview. And so sitting in front of her here is a tray of beads, and she's making necklaces with her fine motor skills at age 100 plus, while she's talking about meeting Amelia Earhart or being the first Black graduate from Grinnell College in Iowa. I think this really represents a redefining of expectations. Aging is unfortunately associated with a lot of stigma, and that stigma really limits a lot of possibilities in aging. I think superagers, as you saw in the video and here in this picture again, Miss June, who loves to travel, and behind her here are some penguins that she's out visiting, gives us an opportunity to change that stigma associated with aging, that it's not always a picture of frail older adults who are not able-bodied. That there's this opportunity to thrive and have great health span even in our later years. So the study of superagers began really more than 15 years ago, and it was centered out of Chicago, where we had the opportunity to enroll mostly Chicago-based participants. And then in 2020, we had the opportunity to submit a grant application that was later funded in 2021 to expand that research. So the initial work really showed us that we could find superagers, that they were out there, and that there was some unique biologic, genetic, and other signatures associated with superagers. In the more recent years, we've been able to expand the study both regionally, now enrolling with our 5 partner sites across the US and Canada. So University of Wisconsin, University of Michigan, Emory University in Atlanta, and then Western University in Canada, also working with a couple other universities in Southwest Ontario, Canada, along with University of Chicago that serves as both the hub site and an enrolling site. And through this work, not only were we able to increase the regional diversity, but also increase the demographic diversity of our cohort, and to layer in deeper questions to ask across this wider cohort. This work is really all rooted in community, thinking about how do we take community engaged research approaches to identify superagers, to partner with them, and with local organizations serving older adults. We've created a SuperAging ambassador board. We get together monthly over Zoom, and they give us ideas about things that are important to them from a research standpoint. But they also talk with us about retention and outreach efforts. And some of them are out in the community sharing their thoughts about the study with others and helping to refer participants into the study. So we really want to support these collaborations and look for opportunities to advocate from a policy standpoint too. So how do we impact that stigma that aging has? There aren't that many studies that really have been enrolling 80-plus-year-olds, especially healthy 80-plus-year-olds. So we think the SuperAging Research Initiative has the opportunity to give good insights about how do you engage a population that's age 80-plus, what might be different than enrolling 80-plus-year-olds compared to 60-year-olds or 50-year-olds or 40-year-olds. So in addition to the biologic, genetic, and other work we're doing, we think superagers give us the opportunity to teach us about the science of outreach, recruitment, and engagement. We have site stakeholders that help us design the study. We have, again, those community and participant stakeholders. We think about ways of giving back. How do we have events, whether they're educational or otherwise, appreciation gifts and materials that might be useful? This picture over here is of a superager who's reviewing one of our brochures, trying to make sure that the language that we put in that is often very technical doesn't make sense to them. Do they understand why it is we may ask them to do a particular brain imaging scan and why that might be useful to future generations and to fill gaps in science? And then we're documenting all of this information so that it can be disseminated and used by others in studies that might be enrolling 80-plus-year-olds or might be generally helpful to either even younger populations. So what is it that we test or ask SuperAging participants to complete? So we have a combination of paper and pencil tests as well as computerized measures. We have questionnaires about family and medical history, brain scans, blood draw, and we have wearable sensors that I'll talk about a little bit later in the presentation here. And then we ask superagers to come back over time. We have these in-person visits every 2 years, but they're also augmented by 6-month visits for wellness checkups and to make sure that we stay engaged with our population and we're not kind of out of sight, out of mind over those 2 years. Brain donation is not required, but we spend a lot of time talking about why it's important, that the opportunity to see cellular and molecular factors that just aren't possible during life, especially within the brain because we don't really regularly biopsy the brain. So this collectively allows us to look at personal and family history factors, personality, psychosocial factors, education, as well as activity. So traditionally, we had looked at these factors via self-report through questionnaires, and then the expansion was to ask superagers to wear wearable sensors during part of their in-clinic or in-research visit when they're here with us, but then also at home in their daily lives. We have robust measures of cognition and then of brain structure, primarily through MR imaging, although some of our participants have had PET imaging. We look at genetic factors, inflammatory factors, as well as immunologic factors. And then for those who donate their brain, we're able to look at those histopathologic or neuropathologic factors. And these are just some of the factors that we're able to collect in superagers. And the idea is to not only have these robust measurements within each domain, but look at the interconnectedness across this multidisciplinary approach. So to date, we've enrolled more than 400 superagers. They join our study. They have to be at least age 80 when they join, but they can join anytime after age 80. So some people call us on their birthday and say, I finally made it to age 80. I'm ready to enroll. While other individuals may not hear about the study until they're 100 years old, and so they're joining then. The current age of our SuperAging participants are from age 80 to age 110. About 60% of the superagers are women. And so I'll preemptively answer the question that might be twirling around in your brain, is that are superagers more likely to be women, or is there a biologic difference? And we can't completely answer that question. We think the 60% probably represents a couple different things. One, that women tend to live a little bit longer, but more importantly, women tend to volunteer for research more. So we have to work a little bit harder in our engagement efforts to identify 80-plus-year-old males to join the study. This kind of sex difference, gender bias we see in superagers is something that is also noted in other older adult studies. So we're not alone in having more women be interested in enrolling in the study than men. It's important to note that the superagers range in education from 6 to 20 years. So it's not that we've just enrolled people who are highly educated. There really is this range. Now, many of them, on average, I think we still bubble around a college education, but we're working hard to make sure we identify people across a wide educational range. And I should mention that each of the superagers, especially for a neuropsychology group, is asked to, we get measurements of IQ. And for each of the comparisons that I'll show you over the next slides and studies, the groups are similar in their estimated IQ. So it's not just that the superagers the differences are driven by differences in IQ. Both the controls and superagers are similar in their distributions. And so this map here represents the locations of superagers that are actively a part of our study. And so you see, although our enrollment sites are only 5 across the US and Canada, there are individuals who move over time, and some of them travel to visit us every 2 years, or we're able to follow them remotely because it's so important to have that longitudinal follow-up. So if we think about superagers, what does it mean to have performance that's at least as good as 50 to 60-year-olds? And so this is just to show you the Ray Auditory Verbal Learning Test is the primary measure we use to differentiate superagers from controls. And an average 80-year-olds are performing here. And then you can see the performance of our superagers, which is at or above what you see for 50 to 60-year-olds. You might then be asking the question, that's great that you found people with great memory performance, but what happens over time? Are they able to maintain that great memory performance? And so we did a study several years ago to look over 2 time points over the period the course of 18 to 24 months. Are individuals able to maintain that memory performance? And we found that they were. And we look across our cohort. For those who have longitudinal data across more than 70 months, we see that this orange line now represents 2/3 of the superagers who are able to maintain memory performance over this time period. So it's not even over the course of just 18 to 24 months, but they actually can maintain this great memory performance for much longer, really contributing to this idea that there's a robust phenotype that is lasting. Another key function or question that we asked early on was, okay, we've got 80-plus-year-olds that have memory like 50 to 60-year-olds, but chronologically they are in their 80s and beyond. So what does their brain structure look like? Does it share more, does it align more closely with those 80-plus-year-olds who they share chronologic age with? Or does it align more closely with those who they share memory performance with? And one of the things that we do in my lab is use FreeSurfer to process structural imaging data. And this gives us the opportunity to label the outer layer of the brain with this, which is depicted in red here, and the gray-white junction, which is just depicted by the yellow line. And then we can measure the distance between these red and yellow lines. And that gives us a measure of cortical thickness, which is a proxy measure for the health of the brain. And so we can do this at the individual level, and we make sure to go through each of these scans to make sure that the labeling that is done in a semi-automated fashion is done accurately. And where there are errors, we go back and correct them, and then re-put them through the pipeline so that we're reducing bias. And then once these are measured at the individual level, they can be put together in groups so that we can compare one group to another. And we get cortical thickness maps like the one shown here, where everything in red and yellow represents areas of thinning in one group relative to another. So when we did this in our superagers, and I want to highlight this is work that's been supported by Dr. Tessa Harrison at the time when she was a research assistant in the lab, Dr. Amanda Cook-Meyer, who at the time was a graduate student, and then Dr. Adam Matterstack, who also has been involved with the lab for many, many years. We saw that when we compare average 80-year-olds to average 50 to 60-year-olds, we see atrophy across the cortex. And that atrophy is on the medial surface as well. And this was not a new finding, but it was this gut check to say, hey, our average 80-year-olds look like average 80-year-olds. The average 50-year-olds look like average 50-year-olds. You see that cortical thinning that happens over time. When we did the same comparison, but now instead of average 80-year-olds, we swap in superagers, the picture is much different. That cortical thinning is absent. And that's because the superagers didn't show any cortical thinning compared to the 50 to 60-year-olds. And then what was even more remarkable is when we looked at the medial surface of the brain, we saw that superagers had a region that was in blue. And blue in this instance represents that superagers' brains were actually thicker than their 50 to 60-year-old counterparts. So this region is in the cingulate cortex, and we know that the cingulate is really important for attention, among other things, and that attention is essential for memory. And this gave us a really important clue of a place to pay attention to, and actually drove some of the early neuropathologic work that we did. And I'll share results from that in just a few minutes. So as we go on, you say, okay, well, that's great. Superagers seem to have kind of youthful brains or youthful cortex. Is that because they were born with bigger brains and they had more to lose, so to speak? Or is it that they're atrophying at a lower pace or rate? And we unfortunately don't have brain scans across the decades of life of superagers, but we do have prospective scans moving forward when they come back at those 2-year intervals. And when we look at the superager brains prospectively over time and compare cortical thinning of average agers and superagers, we see that the atrophy rate of superagers is 2.5 times slower than the average agers. So this really gives evidence that superagers appear to be on a slower biologic trajectory of aging. And we followed this work up by including a 50 to 60-year-old group and really seeing that that atrophy rate that superagers experience over a 2-year period is really indistinguishable and on par with what we see in 50 to 60-year-olds. So lending further support that superagers seem to be kind of shifted in time from a biologic perspective, and their brain aging, if you will, seems to be on par with that of a 50 to 60-year-old as opposed to their 80-plus-year-old peers. And so we've confirmed this result also using convolutional neural nets and machine learning algorithms to calculate brain aging and really get this similar notion that superagers tend to look about 20 to 30 years younger in their brain age relative to their peers. So we've got now superagers with youthful cortical integrity and atrophy rates. And then you may say, okay, well, now what about their white matter? Is everything about superagers more youthful? And so this is work that was done by Dr. Adam Martersek, who is continuing to follow up on this research and looked really at whole brain white matter and hyperintensity and the burden of the white matter hyperintensity burden. And we saw that in this case, white matter hyperintensity burden didn't differ between superagers and controls. So it really seemed that the cortex was the primary area that was more youthful looking and didn't differentiate. And then this result further shows that there didn't seem to be any differences between the males and females in the group. So one of the early studies that we did from a histopathologic standpoint was focusing on this anterior cingulate region. And Dr. Giola at the time was really struck by the number of a special type of neurons called von Economo neurons. And von Economo neurons, if you've never heard of them, I'm going to give a kind of quick overview of them. They are neurons that have only been described in 2 regions of the brain. One of those regions is the anterior cingulate, and then the other is in the frontal insular cortex. They've only been described in higher order species, such as elephants, and they're thought to have something to do with social behaviors. The loss or abnormal development of them has been described in autism, schizophrenia, bipolar disorder, as well as Alzheimer's disease and frontotemporal dementia. They're really large neurons. We typically think of pyramidal neurons or pyramidal cells as being big neurons. And as you see here, the von Economo neurons are substantially larger than what you see in the pyramidal cells. We see that the superagers have 4 to 5 times greater density of von Economo neurons compared to their cognitively average peers. So now, in addition to this brain structure difference, we're now seeing cellular differences in density in the superaging cohort. When we think about doing genetic studies, really most geneticists don't want to talk to you until you have tens of thousands of individuals. But you can ask and answer some questions based on an existing understanding of known genetic risk or protective factors. And one of the ways that we did this was a study in collaboration with Dr. Barbara Spencer and Dr. Sarah Banks at UCSD. And what we looked at was polygenic risk scores for Alzheimer's disease. And the question really was, could you merely identify superagers as people who have extraordinarily low risk for Alzheimer's disease? And would that be is the phenotype just a reflection of really low Alzheimer's risk? And so we compared the superagers on an AD polygenic hazard score to a group of controls from UCSD. And here, the 0 line, I see this is a little bit off center, represents anything above 0 represents risk, anything below 0 represents really low risk. And so for both cohorts, they have low AD risk, but they're not different from each other. If anything, the cognitively healthy controls in this cohort were a little bit lower than even the superagers. We recently repeated this analysis using a slightly different polygenic risk score for Alzheimer's disease. And so now the data are represented as in this plot as individuals in kind of this red color represent those with Alzheimer's disease that was confirmed at autopsy. Green represents, again, cognitively average controls. And then the blue here represents superagers. And we see this almost complete overlap between the controls and superagers, and then this shift to the right for the polygenic risk score for individuals with Alzheimer's disease. So we really replicated the previous result, showing that on average, superagers have low but not unusually low risk for Alzheimer's disease, and that you can't merely identify superagers just by knowing their polygenic risk score. When we think about other lifestyle factors, superagers report, about 80% of them report exercising, 70% report past tobacco use. Now, this is not that we think that tobacco use is good. We think this is likely a reflection of the generation in which these individuals grew up, where smoking was more common. But I think it is important to know these aren't just the individuals who were the non-smokers within that generation. About 80% of superagers report currently drinking. There's really a mix of diet within the group. More than 15% of the superagers are still working. And I really think this is an underestimate of a reflection of how active the superagers are. If they're not working at their primary occupation, they're often volunteering or otherwise contributing to their community and quite busy. In fact, one of the most challenging one of the biggest challenges we have is finding time within their schedule so that they can come spend some time with us to complete their research visit. Another way that we've looked at health was to ask about medication use. And so we compared medication use of superagers to that of their cognitively average 80-plus-year-old peers. And we looked at the number of medications that they use, as well as quantifying the types of medications they used. And what we see is that the medication use is similar between cognitively average 80-plus-year-olds and the superagers. Now, mind you, if we had looked at a population-based sample of 80-plus-year-olds, I think superagers would appear healthier. But it isn't medication use or the types of medication use or types of medications they use that is the differentiator between these two groups. So it's not as if they are healthier overall by that measurement. Superagers also, and this was a study that was done by Dr. Amanda Cookmar back when she was a graduate student in the lab, showing that superagers endorse stronger social connections than their cognitively average peers. Both groups endorse strong social connections, but superagers even more so. This fits really nicely with the larger literature talking about the negative consequences of social isolation and loneliness. And I think gives us something practical as individuals may come to the clinic and ask, what can I do to protect my cognition? What can I do to live long and live well? This is a pretty easy one. Staying connected with others as a priority is something that people can take charge of and take a hold of in their life. But then it's really sparked a lot of interest. So all of these data came from survey data. And so we didn't have the information at the time of what does that mean that they have stronger social connection? Is it that they have really close relationships with a small group of people, or is it that they are the social butterflies of the group? Anecdotally, we see a bit of distribution across, but we're now starting to quantitatively investigate these relationships through the use of wearable sensors. And so I'm going to transition to that part. We asked superagers to wear 3 sensors that are delivered to them via the mail, and they put them on and take them off all by themselves and then ship them back to us. One is on the chest, one is on the wrist, and one is on the ankle. And through there's a process that we're able to fuse these data together to be able to look at activity, to be able to look at autonomic function, sleep, social engagement, as well as motor function. And so this work is led by Dr. Angela Roberts' lab out of Canada. And it gives us the opportunity to quantitatively assess all of these things instead of having just the self-report data. You might be skeptical about, well, is this possible? Will superagers wear them? Well, I'm here to share that they will, and we have greater than 95% protocol compliance and completion of the protocol with really high quality data coming out of it. I can't share too much of that quantitative data yet to date because we're still actively analyzing it, but we see the cohort is highly physically active, and we're able to look at overall volume of data, so the number of hours that you sleep, but also look qualitatively at the pattern. And so these are kind of small patterns here, or small visuals here, but each line going down here is a different day. And this purple part represents when individuals are sleeping. And so you see for this individual who's on the top, they're going to bed about the same time every night, and then they're waking up about the same time every morning, versus this individual who happens to be a control has much more sporadic and erratic sleep schedule with many different and not consistent sleep throughout the night. So bouts of waking and not as healthy of a sleep pattern. And so we're able to dig in deeper than just looking at the number of hours one sleeps, but looking at sleep stages and the distribution of those patterns across the group. And so I want to end there and make sure that there's some time for questions. And just want to thank you for your attention. Again, thank our funders, thank our partners. And if you know a superager, we're still actively enrolling, so we would be happy to have more referrals.
Thank you very much, Dr. Rogalski. But I know that I speak for just about everyone when I say this was a fantastic presentation. Very, very interesting. So we do have a few questions coming in through the chat. The first one we got was concerning cognitive data. Have you encountered any challenges with finding norms for folks that are 80 years old or older? And is anybody in your research lab, are you all looking into providing more normative data for this population?
Yes, this is really, it has been a challenge. So if you look in norm books, there's often, 80+ year olds it's just the category, as if everyone above age 80 is the same, so that there aren't, there are less norms for beyond that age. And then if you look at the ends, sometimes in those 80+year-old ranges, it's an N of 6 or an N of 9, which probably reflects, whoever the grandmothers were in the lab where those data are collected. So I do think that it's a place that we have a great opportunity. And I think there's probably trainees on this call. So if you think about ways that you can contribute, I think this is a gap in the literature, and we have helped to support some of those normative data by providing the cognitively average 80+year-olds. Many of them participated in the norming of the NIH toolbox in a special project dedicated to that. We have the superagers complete the NIH toolbox, but they didn't contribute to those norms because we didn't want to skew them in the wrong direction. But we do think that there is an opportunity to think about ways of improving our understanding of what is normal and what is that expected cognitive ability over time.
And I understand this was largely what the presentation was, but I can't help but think about, how fortunate could I be to ever learn to age like that? Or if there is a way to learn to age like that, I would be fortunate to be able to have that level of cognitive ability later in my life. I've heard a lot about, blue zones and different things that contribute to healthy cognitive aging. One of the questions we had in the chat was if you have any input on what these super agers are eating, if there's any nutrition related information that's coming up.
Yeah, this is a great question. I think nutrition it's certainly important. These are difficult studies to do. Really thinking about, now you've got someone who's lived 8, 9 decades. At what point do we want this nutritional data? And how accurate would it be if we were asking them to think back to, what did you eat growing up? Anecdotally, I will say sometimes I show a video of a gentleman who says, just spontaneously, he's sharing while we're talking to him. Oh, it can't be what I ate. I had way too many TV dinners. And so I don't, I don't share that story to say that diet is not important, but I do share it to say these are not individuals who were all mindful and had a vegetarian or Mediterranean diet. There is kind of our range of our hamburger and French fries lovers to those who have paid close attention to their diet. And it's not that it doesn't matter. I think there's likely more than one path to super aging. Some individuals who probably had genetics that allowed them to eat a few more hamburgers and French fries, where there's others where it really was important to pay closer attention to their diet. And this is one of the things that I think we'll get to understand more as we enroll additional superagers. And so we're really thankful to have this opportunity to enroll 500 new superagers through this multi-site study, and are looking for ways to launch even wider scale studies that are kind of in the works. The diet one is a tricky one to get accurate data, I think, and to really zero in on which part of the lifespan do you want the data from.
That makes a lot of sense. And that's information that I hadn't even considered, that, of course, records probably weren't being kept very meticulously, especially when these folks were children, because who would know that they would become super agers? And then also, like you mentioned, 8, 9, 10 decades, nutrition has changed over time. Speaking of other things that might be contributing to their super aging, somebody else had mentioned whether there may be any history of trauma or other childhood experiences. I'm thinking of like the ACEs study. Do you guys screen for that? And if so, how is that present in the study?
Yeah, I'm really glad that you asked about this. I didn't mention this, but it's a really important point. So you might assume that superagers, you're like, oh, these people probably had life handed to them on a silver platter. I bet these are people who were highly privileged and didn't have anything to worry about, so to speak. That couldn't be further from the truth. Unfortunately, these individuals really represent, when we talk about resilience with superagers, one of the lenses and ways that we've looked at it is through life experiences. So we have superagers where they are the only survivor in their family of the Holocaust. So they themselves were sent to a concentration camp, and they were the only ones who survived. We have other individuals who have experienced trauma in the form of losing a child or a sibling at a young age, individuals who were born into poverty and stayed in poverty throughout their life. And certainly that's not the whole live experience of the entire cohort, but I would say that there's many individuals who have encountered hardships and challenges in life, and I think it speaks to their resilience that they had an opportunity many times across their lives to say, gosh, things are too hard. But they didn't do that, and they really have looked for the good. And so we've collected data in the past that we're not finished analyzing about life stories of superagers. How do they share their story? And when they describe, we give them a prompt to describe a challenge in their life. Well, when they tell that story about a challenge, do they continue to talk about all of the bad things that happened thereafter? Or do they say, gosh, this bad thing happened to me, and then I learned so much from it, or this good thing came out of it? And so these are data that we're really interested in saying, is it their perspective taking that helps them with their resilience? And these things are important because they represent, again, other opportunities for intervention and reframing.
It sounds like, obviously we don't have the solid answer yet of what causes super aging. How do we get there? There are so many different pieces, and I'm very excited to read that study on the different stories of these super agers, and especially how they manage the resilience. Somebody else had asked the question about language proficiency. So some research is out there to suggest that bilingual folks and people with even more language tend to have higher cognitive reserves. So than folks that only have one language. Was language fluency assessed, and was that something that showed up significantly for you all?
We do ask about the number of languages individuals know or use on a regular basis. I don't have the percentage off the top of my head of how many individuals are bilingual or know or are fluent in another language. I can say anecdotally, I can think of people who within the cohort where that has been a priority, but certainly many others that's not the case. One particular individual who that's one of her primary activities is still learning new languages. So she's actively engaging in that. And so I think that really speaks to work that was done by Denise Park years ago, not just on like language acquisition, but she did a really lovely study years ago where she had 3 groups of people. One group that learned to knit, another group that took a photography class, and then a third group that I think watched TV or something similarly non-active. And the question was like, where's there the most brain benefit? Is it in learning to knit or photography? And I think you could kind of swap in learning a new language. And in this case, what she showed was that both knitting and photography showed similar brain benefit. And it was this idea of engaging in new learning and challenging yourself that seemed to be really key. And so we've tried to focus in on asking some questions of how are they staying cognitively active and where that might play a role.
These are all very helpful. So I realize that we're getting low on time, so I'm gonna try to get through some of the questions a little bit more rapidly. But do you happen to recall, even if you don't have the exact figures, do you happen to recall if there have been any superagers that have a higher than typical risk of Alzheimer's or any other dementia processes that didn't tend to develop them?
Yeah, this is a great question, and something that we're actually actively summarizing right now. We do have individuals we have a few individuals who have extraordinarily high risk. And so they are particularly interesting to understand how is it that they are able to tolerate such high risk and still maintain great cognitive performance over time. So stay tuned for likely a case series or a couple case reports that comes out of that. And luckily, we've had the opportunity to do whole exome sequencing, to layer in both genetic side of things, as well as our really robust cognitive battery. And so you can kind of think of these as opposite end of the spectrum. There are some people who have an E2 allele, although we don't see that's thought to be protective, ApoE2 versus those with ApoE4 or have a high polygenic risk score. And so if in one group, you can think of them as having some of these protective factors, and this other group really having this accelerated risk. What layers on top of that allows them to mitigate? It's not dissimilar to the idea that we see with the autosomal dominant escapees in the Diane cohort, where that allowed for the discovery of the ApoE, the Christchurch mutation. So what is it that might be layered on top from a genetic standpoint that allows someone who otherwise seemingly has high risk, but it doesn't manifest in the same way? So these actually in individual case studies, even if it's not a high percentage of people, can be extraordinarily helpful for showing what's possible.
And somewhat similar to our theme of questioning so far, I know once again there's no one answer, but we're still pushing for what are some of those factors that might be contributing to this super aging. Have you seen any major cultural differences towards that resilience, or whether that's emotional or cognitive?
Yeah, and so when we think about cultural perspectives, this is where the expansion of the study became even more important, is because primarily we were recruiting within the Chicagoland area, so we were getting perspectives of individuals who at least regionally were living in the same place, even though they may have come from different walks and styles of life. Unfortunately, we have not done a nationally representative cohort yet, although the expansion across 5 sites gives us the opportunity to have quite a bit more diversity than we had. So I don't have a a good quantitative answer for this yet, but I think we are getting more robust perspectives through the expansion of the study.
Very exciting. And I know myself and I'm sure everyone else here is looking forward to the expansion of this study and how many more directions this is going to go. And yeah, we're already very excited about the data that's being presented, and we can't wait to learn more. So I think we actually got through all of our questions right on time. So I just want to thank you again, Dr. Rogalski, for this excellent presentation. We really sincerely appreciate it.
I appreciate you having me.
Yes, of course. Whoops, there we go. And I just want to remind everybody that we have another lecture next week. So please join us again for another lecture on diversity factors in neurorehabilitation, assessment, and participation with Drs. Wisinger and Eiley. Thank you all for your attention, and thank you all for your attendance. And we look forward to seeing you again soon. Thank you.